I'm going to do something a little different this year, and we're bringing this down from research, which is what drives all of the innovation of everything that we do and why we raise money. But I'm always asked to talk about either basic genetics, so that we can kind of bring this to a level where, if we talk about the word gene, we've seen that a few times, we understand what that means. And I'm going to focus a little bit on inheritance patterns today, because I usually never have enough time to get there in my basic genetics talks. So forgive me for not having as much audience participation as we usually have, where everyone's a codon and we're making a protein. There's videos on that, so I wanted to make sure that I point everyone to those videos that are on the SPF website. And we can review that too, and I'm happy to answer questions about that. But I'm going to jump a little bit beyond that to specifically talk about inheritance patterns. But like I always say, I want this to be interactive. This is ideally, even material that's taught in high school now in a lot of cases. And I've been talking with a couple of high schoolers here even, and that's confirmed. And my kids are learning that. So some of the stuff that I was learning as a freshman and even sophomore in college is often down in the high school level, but that's not necessarily the case with everyone in this room. So the idea is to make sure if you have questions, let me know. And a lot of this is information that you'll find out as part of your diagnostic odyssey, and I'm sure for a lot of you it's been an odyssey with some dead ends, trying to figure out how to understand the situation that you're in, but you've ended up in this room. So that means that you're in a better place along that diagnostic odyssey than you've been in the past. But there will be support that is available for you today in these areas, especially talking about inheritance. You've got genetic counselors, and actually some of these slides have come from great genetic counselors. You'll have genetic counselors probably in most of the hospitals or physician offices that you're part of, and also some of our sponsors here, including Invitae and MNG, will certainly have excellent genetic counselors at your disposal that you can call up and get a lot of this information to, and they'll walk you through it in a personalized way. But I thought that I would go through some of this basic genetics information because it's just good to understand. It's good to understand what's going on in your family. It's good to understand what the risks are of passing a lot of this on to the next generation and how it can look at as a whole. So we'll go through some specific examples, and I will ask for audience participation, just not standing up in front. So we will get to that. But I want questions. So as you have questions, either write them down or raise your hand, and I'll come out. I'll reiterate the questions for the camera so that we can make sure that people that are watching the video afterwards can get this. Can I count on you to participate? All right, excellent. So I'll start a little bit with talking about the fact that just about all diseases have a genetic component. And it's not equally the case for diseases for which there is a significant environmental impact. And you can imagine that's easily the case with something like cancer. If you're a smoker, it makes sense, right? I mean, 40 years ago, we might not have seen, oh, that's going to give you risk of lung cancer. But we know now that that's the case. So in some cases, there are complex diseases that actually have a combination of environment and genes. There are some diseases for which environment is most of the contributing factors. But there is a whole class of diseases that we call Mendelian diseases. And you might remember this comes from the name Gregor Mendel's, kind of like the original father of genetics, who did all of his pea pod experiments and pea plants. But they're called Mendelian diseases, and there's a couple of examples listed here. You may have heard of Huntington's disease as a neurological disorder. We talked a little bit about spinal muscular atrophy earlier. That's an example too. Those are two extremely devastating diseases that can be terminal. And that's luckily not always the case with our diseases in this room. CMT is another neurological disease. Of course, cancer, there are some significant forms of inherited cancers, but it's that cancer disease, which obviously is lots of different types of cancers. It can also be a complex disease where there is a significant environmental impact, like smoking, for instance. So some are in both categories, but HSP is in the category of Mendelian diseases because there are a lot of very well-known genetic causes. There's fewer genetic causes known for PLS, but there's a lot of emphasis, and we actually fund a lot of work to move into that area to identify what those genetic causes might be. And that's some of the work that Sabrina and Christine were talking about earlier. Questions? So if we look at Mendelian diseases specifically, these tend overall to be quite rare. We got a great overview of rare diseases from Jeffrey earlier about, I mean, we're talking about 7,000, 8,000 different rare diseases, although individually they add up to about, 1 in 10 people in the world has a rare disease. I mean, that's phenomenal, right? That's an incredible recognition. I'm not sure that was necessarily recognition until quite recently, but so all of these rare things added up become quite common when taken all together. You can have genetic change in one gene that causes disease, and you've heard that some of these diseases can be quite heterogeneous. You can actually have, in the case of HSP, well over 60, and I think there's probably 150 or, and Dr. Fink's going to talk later, he usually gives really recent updates on how many different genes are known to be associated with disease. We're routinely sequencing, and in Vitae and MNG, for instance, sequence dozens. Upwards of 60, even more genes today. You can order that test, and it costs less than it ever has before. But in each of these cases, each of those one gene cause disease. They're all linked to be causal. Inheritance patterns for these genes are well known and easily predicted, and I'm going to be using a lot of words that I want you very comfortable with, as we'll walk through this. And if you have questions, please raise your hand. Autosomal dominant, there's a couple of examples of those. Huntington's disease, and in our case, SPG4 or spastin is the most common form by far of HSP. It's responsible for about 40% of cases, even in people in this room, about 40% of people that have HSP have an issue with the SPG4 or spastin gene, okay? SPG3A is next at lastin, about 10% more, and then each gene contributes less than that, adding up to probably 150 or so genes, maybe more. Of those genes, there are many, including SPG4, that are called autosomal dominant. And I'll tell you what autosomal means. I'll tell you what dominant means as we go through. There's autosomal recessive. Some examples of that you might have heard of outside of HSP include cystic fibrosis. Who's heard of cystic fibrosis in this room? Most people have heard of it in some way because there have been advances in genetic forms of therapies actually recently with cystic fibrosis, but it's more common. It's more common than some of the diseases that we're talking about here. SPG7, paraplegia, is an example of an autosomal recessive disease in our area, and I'll go through a few others as we go through. As opposed to autosomal, there's X-linked dominant. So when I say autosomal, who knows what I mean by autosomal when I say that word? Very few. Okay, that's good. That means I'm talking on the right topic if we're not sure. So how many chromosomes do humans have? Does anybody know? In our body, in each one of our cells, there's DNA in each one of our cells. And remember, DNA is the thing that goes from generation to generation. DNA is the blueprint for making us what we are. That's what gets transmitted through sperm and eggs to the next generation. One from mom, one from dad, right? That's the only information that goes. And remember, DNA is what makes proteins, and proteins are really those building blocks, the 2x4s, the structure, like the carpet on the floor here, the cell membrane, the things that hold it together are proteins, but they're all encoded by the DNA. It's the DNA that gets transmitted. Does that make sense? If you think back to Jurassic Park, right? They're taken, I mean, that's fiction, but in some ways it's science fiction based somewhat in reality. It's the DNA that is so stable, it can actually survive in some cases for many, many thousands, even millions of years. It's a very stable thing. So it's a great thing that can be transmitted from generation to generation. It's a very stable, good template. When I'm talking about autosomal, I'm talking about chromosomes, and I had asked a question, but I didn't hear an answer. How many chromosomes do humans have? 20. I heard 21. I heard 26. You're getting close. Somebody said 23. 23 pair. Have you heard of 23andMe, a company? Well, there you go, right? So there are 22 chromosomes plus a pair of sex chromosomes, okay? So when I'm talking about autosomes, I mean those 22 pairs of chromosomes that are not the sex chromosomes. What determines if you're a boy or a girl when you're born is if you have two X chromosomes, you are a girl. If you have an X and a Y chromosome, you are a? All right, so those are the X-linked or sex chromosome diseases. So when I say X-linked dominant, I'm talking about genes that are on the X chromosome. The X chromosome, girls have 2, boys have 1. And we'll talk about that a little bit later. When I'm talking about autosomal, so what does that mean? If it's an autosomal gene, what does that tell you? It's on an autosome. Where is it? I heard it. It's not sex-linked. It's on one of the other 22 pairs of chromosomes. It might be on chromosome 1. Chromosome 1 tends to be the longest. 2 is the next. 3, not exactly down in order, but they tend to be in order. Some chromosomes are very long. Some chromosomes are very short. But almost every single cell in your body has a complete set of those chromosomes. All right? And obviously that's what gets passed on to the next generation. Do I have you all already? Okay, I don't want to go too fast. So when I'm talking about autosomal dominant diseases, it means that we've got a gene that's on an autosome, one of those 22. If I'm talking about X-linked, it means that the gene is located on the X chromosome. It's one or the other. I will at the very end, because whenever I say always in genetics, there's also always an exception, sorry. But in general, your genes are on those locations. I'll talk at the very end about mitochondrial diseases, because there's a little bit of DNA that's a mitochondria. We'll worry about that at the very end if we can get to it. X-linked recessive we'll go through. There's a couple of examples too, okay? And in these cases, in Mendelian diseases, the environment and exposures to different environmental conditions have very little impact on the disease, progression of the disease. There's always some. Again, it's never always. But in this case, it's not about whether you smoke or whether you don't smoke. That has other consequences, but that's not necessarily going to have a consequence if you have a Mendelian disorder. Make sense? Okay. All right. So I just went through classifications, right? Autosomal versus X-linked. So the main question, and now who knows the answer to this question? What does autosomal mean? Does everybody know? Raise your hand if you know what that means now. All right, so we've all learned something. That's a good thing. So is the gene on a chromosome that determines sex? Now I'm going to talk about dominant versus recessive. If you're talking about a dominant gene, and this is a fundamental piece of information about just about every one of the genes that we know about, and we've cataloged the genes, right? This happened back with the Human Genome Sequencing Project. We've cataloged as many as we can. We might not know everything about those genes. We continue to learn about those things, but we've been able to catalog them. We're probably missing information. We certainly know that there's stuff that we haven't been able to interpret, but we're on an interpretation mission now. One of those things that we want to understand is it a dominant gene? Is it a recessive gene? Sometimes it gets a little more complicated, but almost always it's one of those two flavors. And that's the case with the long list of 60 or so genes that cause HSP. We know which ones are dominant. We know which ones are recessive. I actually pulled up on my computer just a few minutes ago a gene review that is written by our esteemed Dr. Fink most recently that lists out all those genes. It's literally a quick Google search away. Search for HSP, gene reviews pops right up, and you can see, oh, here's all the genes, and here's the table for dominant, here's the table for recessive. It's all at your fingertips. And now that you know these words, it's not necessarily as much of a mystery, right? So what matters when you're thinking about dominant versus recessive is how many copies of the gene are necessary to cause the phenotype. What that means is, do you need one mutated copy to get the disease, or do you need both of your copies? And when I say one or both, do you know what I'm talking about? Because you're getting one full copy from mom, you're getting one copy from dad. Remember I said sperm and eggs come together, and that is what gives you your complete genomic makeup. You're getting one copy of all 22 plus a sex chromosome from your mom, one from your dad. So you're getting a full repertoire of all genes from your mom, all genes from your dad. If you have a dominant gene that's inherited from one of your parents, you're going to have disease. That's what that means. How many copies of that disease gene do you need in order to show that phenotype? And I'm going to go through that in a lot of detail, okay? So that's the main question, is how many copies are needed? And I'm going to go to the autosomal recessive situation is a little bit more complicated, so I'm going to start that first, and then dominant's going to be a piece of cake. So in this case, both alleles, when I say alleles, I mean a copy of a gene, one copy of a gene, It's either going to be maternal or paternal. You might not know, but one came from mom, one came from dad. And in this case, both alleles in a gene pair must contain the pathogenic mutation. I don't want to go too fast, so I'll let those words sink in, all right? Both alleles in a gene pair must contain a pathogenic mutation. That means in order for a recessive disease, for you to have a recessive disease, you have to have a pathogenic gene from both mom and dad. You can imagine that's a pretty rare scenario, okay? But it happens. It's happening in this room, and it happens all over the world, and I'll talk more about that. Individuals with one allele are carriers and are not affected by the condition. So if you have only one copy, either from mom or dad, you're not going to show a phenotype at all. You're going to be normal with respect to that disease. Does that make sense? Everyone follow? So you need 2 copies to actually have disease. If you only have 1 copy, you're called a carrier. What if you have 0 copies? What is that? Normal. I heard normal. Perfect. Exactly right, right? And you have 0 chance of passing it on, okay? And we'll go through some of those. So if you think about the different scenarios of inheriting a mutation from mom and dad, you can imagine, and we'll go through the math, that 25% of your offspring, when both parents are carriers, have a risk of having disease. And we'll go through how that math works. In this case, males and females are equally affected. Why? Why are males and females equally affected when we're talking about autosomal diseases? Because it's not sex-linked, okay? So it's not going to be different if you're a boy or a girl. It's going to be exactly the same for both genders, okay? So some examples in neurology of recessive diseases is I heard, I think Christine talk about SMA, and I talked about that a little bit yesterday with CRISPR, and it's so exciting because there is the new therapy, Spinraza, nusinersen, that was approved just this past year, and it's a motor neuron disease with a therapy. Holy crap, right? That's pretty awesome. That's really good news for this audience. It doesn't mean that that's going to help us directly, but the fact that there's a therapy for motor neuron disease should give, I think, this community a lot of power, and it's certainly risen my spirits quite a lot. If I have time, I can get into that a little bit more. Friedreich's ataxia is one of the most common forms of ataxia, and that's an autosomal recessive disease as well. Examples in the world of hereditary spastic paraplegia include SPG5A, and those of you that know your subtype, you might find this familiar. SPG7, which is the protein's called paraplegian. SPG11, which is called spataxin. SPG35, FA2H, I actually saw that out on the question board, so I wanted to make sure that made it on the slide. Those are examples of autosomal recessive conditions. Now, I just went through this, so this should look pretty familiar. If you have no copies of a mutated gene, you're going to be healthy, you're going to appear healthy, and you're not at risk of passing that on to your offspring. Remember, you got 1 copy from mom, you got 1 copy from dad, and the same thing happens with the next generation. You're going to donate 1 of those copies, independently assorted. You can't pick which. You're going to give 1 to your next generation, whether it's going to be a boy, girl, whatever, right? That's what's going to get passed on. So you have 0 risk of passing on a disease gene if both copies are normal. In the case of recessive diseases, if you have 1 gene that's mutated, you're going to be healthy, but you're going to be a carrier. And what does carrier mean? I just kind of went through it a little bit, but what does carrier mean? Just to make sure you're along with me. I'm hearing risk of passing it on. That's what I'm generally hearing. Exactly right, right? You are not going to have phenotype. Maybe you'll have some very mild phenotypes because, like I said, you never say always. But for the most part, you're not going to have disease. right? But you do have risk of passing that on. So carrier status means that you've got that risk of passing on the next generation. And that's where, that's what I really wanted to talk to you guys about, so that you feel comfortable going into, for instance, a genetic counselor's office, empowered with that information. So you can worry about other details, not necessarily this big picture of, do I have risk of passing on or not? You can think about details related specifically to your case, right? So I want to give you some basic information there. Now, if you've got 2 knocked out copies, that means that you're going to present with disease. And here are some examples of people that are carriers of autosomal recessive conditions. Everyone in the world are carriers of recessive disease. Actually, between 7, 8, 9, 10, 12 different diseases. Okay? Everyone in the world. It's just a fact. There are frequencies, carrier frequencies, for all these 8,000 different conditions plus, such that you are certain to carry half a dozen, maybe a dozen different genetic conditions, and you'll never know. And until we started being able to sequence people's genomes, we didn't really know that. We thought it was theoretically the case, but we see it now. If you sequence your genome, you will find them. Now, that can cause apprehension in some people, right? So we don't necessarily recommend that clinically or medically to do that yet, because the last thing you want to do is cause undue stress and strain. It's just a fact, and you don't have to worry about it. That's normal. That's perfectly normal. So if you look at, who's this? Who's the guy on the far left-hand side? Is it Boomer? Yeah, it almost looks like Phil Simms too, right? They look kind of similar, and I think that at least in the case of Phil Simms, he's got a kid that's now a famous quarterback as well, right? So this is, I would say that Boomer and Phil are renowned for having some pretty amazing genes, right? Athletic ability, but certainly they are both carriers for a dozen recessive diseases. Totally normal, all right? Maybe even Beyonce. So if we look, this is something that folks that have taken any genetics class would be quite familiar with. It's called a Punnett Square, but it's a very good way to understand more about how you can calculate risk of passing on and actually having disease. It's a good visual way. And this is where I'm going to ask you guys for audience participation later to quickly generate your own Punnett squares. By the end of this, you're going to be able to do that. Piece of cake. Just scribble even in the corner, look at it and say, oh, I understand that risk in my family and somebody else's family, reading about it, and I want you comfortable after this talk being able to do that in the corner of a piece of paper. Okay, that's my goal. And if you can't, let me know, because I want to be able to get you there if that's what you want. So if you look, for instance, at Dad up top, and you can't all see my pointer, I'm sorry. I'm looking, let me try to do this. Right here, can you see my arrow? All right, so here's Father. Remember, you've got 2 full sets of copies of genes, one that came from your dad, one that came from your mom. And if you look at the risk of a father passing on one set of chromosomes or another, that's what this indicates, right? He's got a chance to pass on this chromosome, wherever it is. Remember, this is autosomal. It's one of the 22 chromosomes, not the sex chromosomes. If you look at this chromosome, where is it? Look at this chromosome. That is a normal chromosome. If you look on this one, it's indicating that there is a mutation, right? So consider that your gene, right? So if you graphically map it out, he has a chance of passing this one off, and what's his chance of passing off that chromosome? He can pass off one of 2. What's his chance of passing off one? 50%, exactly. So there's a 50% chance that he's going to pass along this chromosome, 50% chance of this. Here's Mom on the other side of the Punnett Square. She's got a 50% chance of passing on this chromosome, 50% chance of passing on this one. So if you literally cross, and I'm not talking about, artificial breeding here. I'm talking about hypothetical combinations of this chromosomes. You can have 4 different combinations, right? You can have, and I'm talking about 4 different combinations of children. You can have a baby in this quadrant that has this chromosome from dad and this chromosome from mom. And you can have a baby and this is each of these quadrants is exactly 25% likely, because there's 4 different possible combinations, right? So you can have a baby here that has this chromosome from dad and this chromosome from mom. And same way, in this quadrant, you can have a baby that has this chromosome from mom, or this chromosome from dad, this chromosome from mom, and same here, okay? And if you just draw that Punnett Square out and just mark it off and interpret the results, you can see that, for instance, there is a 25% chance of having a baby with a genetic condition. Does that make sense to everyone? If that doesn't make sense, you don't have to raise your hand if you're not comfortable, but I want to kind of gauge. Does that not make sense to somebody right now? Because I want to make sure I go through this slow enough. Because we're going to be able to build from here. If not, you can ask me afterwards. So so for instance, if you had this couple that had 4 children, what are the chances that one of those children is going to have the genetic condition? 25%. Is it possible that 2 of the children could be affected? Why? 25, oh man, you guys are good. I heard 25% chance for each child. So it's like flipping a coin, right? If it's 50-50, you can get 4 heads in a row. It's unlikely, but the next time you flip, it's not more likely to be tails. It's still a 50-50 chance of being heads or tails. Exactly. So if this couple had 4 children, all 4 could have disease. It's unlikely, but it could happen. Each child, you have an independent risk, 25% risk of having a child with genetic condition. Okay, so more complicated. What's the percentage of children from this combination that will be healthy? Healthy children. What is it? I heard 25%, but it's not 25%. It's 75%. I'm talking about healthy children. Now, what percentage are carriers? 50%, exactly right. And those carriers are healthy in this case. Because remember, with a recessive condition, you need 2 mutated genes to show disease. Make sense? So that was a trick question, sorry. But that's okay. That's why we wanted to go through these slides, because it is kind of tricky and it can seem confusing, but it's for a darn good reason. I don't want this to be a black box to anybody. Okay, so with this combination, I don't want to dwell on it much longer. I want to be able to move on, but we've got, out of a complete set of children you can have, about 25% will have disease, about 75% will be healthy, about 50% will be carriers. Make sense? All right. So let's move on to autosomal dominant. This is an easier situation. How many copies of a disease gene do you need to have an autosomal dominant disease? 1. 1 allele in the gene pair contains what in this case is called a polymorphism mutation. I distinguish a difference between those, but it's a change. It's a change from normal. All individuals with a mutation are affected by or predisposed to developing the condition, and you have a 50% reoccurrence risk for all offspring. I'm going to prove that to you with the same Punnett Square. I want you to be able to prove it to yourself as well. We'll show why that's the case. Males and females are equally affected. Why? Because it's not on the sex chromosome. We're talking about autosomes. Good. Examples in neurology is the devastating disease, Huntington's disease, tuberous sclerosis, TSC, many forms of hereditary spastic paraplegia, including some of the most common forms, SPG4 or spastin, SPG3A, elastin, SPG6, NEPA1, SPG31, REPA1. So those are some of the most common forms. The list goes on quite long. Most of these common diseases are autosomal dominant. So again, mutations in both genes can cause disease. If you've got 2 copies that are normal, you are healthy. If you have 1 copy that's mutated, you have disease. And in this case, there's actually a picture that shows if you do, like in recessive, if you actually inherit both copies, that is a disease phenotype. I would say sometimes it can go even further and say that's not necessarily consistent with a live birth. And you might not even know, right? There might actually be a spontaneous abortion you might not even know about, or loss of baby early on in some cases. I'm not sure if that's the case I don't see very many combinations of autosomal dominant HSP. I don't see many where you've actually got double knockout. So sometimes that's consistent with disease, but often it may not be consistent with life either. Autosomal dominant, continued again. Here's your Punnett Square. So dad has the genetic condition, which you can easily imagine. He's going to pass along one chromosome or the other. In this case, mom is unaffected, does not have any copies of a mutated gene. So you've got that 25% chance of 4 different quadrants. So in this, oops, sorry, I went ahead by accident. In this case, how many, what's the frequency of a healthy child? 50%, right? Because 50% of children have no inherited mutated gene. What's the chance of having disease? And again, prove to yourself, does it have anything to do with whether you're a boy or a girl? An autosomal? No, not the case, right? Are there any carriers? Why? Why no carriers? Because if you have one copy, you have disease. There's no carriers. In some ways, you can think of people that have disease, autosomal disease, as being a carrier because they're actually going to pass it on, but really a carrier is a term that's only used for recessive. Does that make sense? So with autosomal disease, if you have a copy, you have it. What's your chance of passing it on? Sorry? 50%, exactly, 50%. And I've heard a lot of us say that commonly, but you might not have understood why, right? It's just kind of ingrained, you know it, you've read about it, this is why. Is that informative? Okay. We'll get a little bit more complicated, but this is important to understand. Penetrance. Remember I said nothing's ever 100%? Here's one of those shades of gray, okay? Penetrance is the chance that an individual expresses a phenotype given their genotype. So a penetrance of 70% means 70% of people that have that genotype, that have that disease, will show a phenotype, okay? So maybe you've got variability within your family. Who has variability of disease within their family? Pretty common with an HSP, right? In some diseases, there isn't much variability. In most, there's at least some variability. In some cases here, it depends upon the gene. Some genes have more penetrance than others, and so you see some of that variability. Another way that you can have variability is with a term called expressivity. And if you're talking with a geneticist, they're going to use penetrance and expressivity pretty commonly, maybe not in front of patients because they don't want to talk about something that's not widely understood, but these are pretty common terms. And in some ways, it's just showing shades of gray. So expressivity is the extent to which a disorder is expressed as a phenotype, but somewhat different from penetrance, right? But same kind of idea. If a disorder shows variable expression, then affected individuals may express a different collection of possible syndrome symptoms for that disorder. So common disorders are like multiple organ systems and so forth. Questions about that? If not, I'll keep moving on. All right. Anticipation. I want to make sure that you know anticipation is not common in HSP. And I'd love for Sabrina or Christine or John to correct me if I'm wrong about that. But I don't believe that's much a case. But I do want to talk about it because it's something that you'll hear about, and I don't want you too freaked out about it. So in some cases, you can actually have a mutation that worsens in successive generations. That's not the case in a lot of the diseases that we're talking about, but if you're thinking about something like Huntington's disease, where it's actually caused by an expansion, not just a single change, you can imagine, and I've used examples before, and you can review a video on it, where you can actually have a lengthening of this repeat region, and that's associated with a worse phenotype, and then if in the next generation you have further expansion, that can be even worse. And so in some diseases, it's very important and well characterized, but not with most of ours. So for instance, in the case of myotonic dystrophy, again, this is a repeat disease. It's with the genetics and how the genetics work and how it's likely to expand or contract within a repeat. You can have a primary generation that has cataracts, and maybe the next generation that has grip myotonia. You always know if you shake the hand of somebody with myotonia because they have a hard time letting go. It's just the nature of that disease, right? bilateral cataracts or facial weakness, a worsening of progression. And then the next generation, perhaps even much more severe than that. So that is a genetic phenomenon, but not highly relevant to this population. So I'm not going to dwell on it much more. I don't want people to worry inappropriately. Talk a little bit about X-linked inheritance and see how I'm doing on time. How are we looking for time? Okay, I'll try to go a little bit quicker because I want to leave plenty of time for questions. So there's two kinds of X-linked inheritance, but really I'm going to be talking about recessive, not necessarily dominant, but I'll give a few examples of both. So if we're looking at X-linked, now it will matter if you're boy or girl, right? You may have different risks, and we'll talk about how that's the case. So males, again, have one X chromosome and one Y chromosome. Y chromosome has very few genes on it because girls don't have a Y chromosome, right? If there was an essential gene on the Y chromosome, girls wouldn't have it. That doesn't make a lot of sense, right? So basically, the genes that are on there tend to be specific to males and generate those things that are male-specific. And in some ways, you can consider males as having less genetic material because they only have one X chromosome, whereas girls have two. And so a recessive condition is going to show up in a male, finish my sentence. If you only have one X chromosome, and even if it's a recessive disease, if you're a girl, you're going to have more genetic material to actually be able to compensate for the loss. So you actually need two copies in a girl, but if you only have one copy of the X chromosome when you're a boy, you're going to have the disease even if you only have it on your one X chromosome. Does that make sense? All right, we'll go through how that actually looks. So the disorder is expressed in males because they only have one X chromosome. All right, Duchenne's muscular dystrophy is an example of this. It's mostly in boys. If you have girls, it's a lot less likely. And it's because males only have the one X chromosome, okay? There's no spare X chromosome to compensate like women. In this way, for X-linked diseases, women are genetically much stronger, right? That's just a fact. So examples of this are muscular dystrophy, some very common forms of color blindness. I have a couple of boys that are both colorblind, and it's X-linked. And I worry as I start to teach them how to drive, they can't distinguish green from red. Does that seem like a problem? Could be a problem, right? Yeah, I'm thinking about that. It's occupying more of my thought. HSPs including SPG1, SPG2, 16, 22, those are examples of X-linked recessive, and that may be on your list. Okay? So, Punnett Square time. This one's more complicated because X versus Y means boys and girls. So if we've got, and I'll use my little pointer here, is this at the top the father or the mother? Father. Father's going to donate 50% of the time an X, 50% of the time a Y. Remember, it's what the father passes on is what determines the sex of the child. If they pass along a Y chromosome, the child's going to be a boy. If they pass along the X, you can only get an X chromosome from mom, so you're going to get an XX in this combination, XY, and XY means boy or girl? Boy. And in this quadrant, girl, in this quadrant, boy. So we all know you have a 50% chance of having a boy, 50% chance of having a girl. This kind of documents that out. Now, if we're looking at this X chromosome being affected, which means the father, in this case, is probably showing disease, what are the chances of having an affected daughter? Trick question. I'm going to fill this out for you. You could have filled this out in the corner of your paper as well, and I'd recommend that you start to get good at that if you want to. Practice it, because it's easy to do. It's easy to just make a little square, have your X and Y on top, have some sort of demarcation for a chromosome that has a mutation on it, and look at the combinations. So the question here is, what are the odds of having an affected daughter? Look at the daughter quadrants, the XX quadrants. We're talking about recessive disease. What's your answer? Zero. I've seen 0. Are there other answers? Does everybody agree? That's exactly right. There is a 0 chance of having an affected daughter. Is that important? That's pretty important, right? That's important to know. And it's not necessarily intuitive. So from this parent, you're not going to have any affected daughters. What's the chance of having an affected son? 0. Is that important too? Wow. Okay, there you go. What's the chance of having an affected, or I'm sorry, a carrier child? What's that? A carrier child overall. So looking at all 4 quadrants, 50%, exactly right. What's the chance of having a carrier daughter? 100%. Every single daughter will be a carrier. That was the case with color blindness in my case. It came from my wife's side, and she got it from her dad. This exact same scenario. We know she's a carrier because she got it from her dad, 100% chance. Now, to pass that on to my sons, if we flip these around, if she's, and I think I might have that as the next example, X-linked recessive. All right, so here's where the mom is a carrier. She might not know it. My wife isn't colorblind. You might have the same kind of situations in this room, right? And I am not a carrier. I didn't know that, but I don't have color blindness, and that would have shown up. So what are the chances here of having an affected daughter? Again, feel free to scribble in the corner. So what are the chances of having an affected daughter? 0. Exactly right. Color blindness is very rare in women, and this is why. What are the chances of having an affected son? It's recessive. You're exactly right. But you only have 1 copy of the X chromosome, and so you don't have that other copy to actually have a normal. So in the case of X-linked gene, you only need 1 if you're a boy. So that you're going to have a 50% chance of having disease in this case. So an affected son, 50%. I've only got 2 sons, but they both have it, right? Independent of sort, you flip that coin, you might get heads twice. It's just the way that it is. That was the case here, maybe the case with folks in this room. What are the chances of having an affected child overall? 25%, exactly right, right? Useful information to know? And this is the sort of thing that, like I told you, a genetic counselor is going to be able to walk you through these scenarios for your family, but you can also do a lot of this math yourself. And obviously, I mean, I'm at your beck and call too. I mean, if you need anything from me, I'm not a clinician, right? I'm a lab geek. I'm into the genetic side. I'm not going to give you medical advice, but I can walk you through these scenarios, and it's really important to talk with folks like genetic counselors about these kinds of things. All right, anyway. What about X-linked dominant? I'm not going to spend very much time on this. What are the odds of having an affected daughter in this case? Now, with dominant, can girls be affected now? Yes, right? Because you only need 1 copy and you're actually going to show disease. So what are the chances of having an affected daughter? I don't know of any HSP, by the way, that's X-linked dominant, just to let you know. But there are a few cases of this in the world, so I want to go through the theoretical possibilities. What are the chances of an affected daughter? In this case, affected daughters, 100%, exactly right. What about an affected son? And this is because this is an affected male, which is not really a very likely scenario in this case, but that's okay. Affected son in this case, 0. Yeah, that's what I heard. An affected or a carrier child? Are there carriers in dominant? You can actually have, in this case, you've actually got risk of your girls passing it on, not your boys. But yeah, so there's actually a carrier doesn't have, it's kind of a trick question, right? It doesn't really have meaning in this case. No, X-linked cases are only recessive that I know of. There are several genes that are X-linked, and they're all considered recessive, not X-linked dominant. That's what I intended to say if I wasn't clear. Does that make sense to everyone? I'm realizing that's the first question too, which means I'm not doing my job. So that means that there's more and I'm not getting them. So let me zip through this a little quicker. The one exception, remember I said before, is there's autosomal and there's X-linked, and there's one more thing, mitochondrial. And I don't want to dwell on this too much, but there is one example within HSP, It's a mitochondrial gene called ATP6. I think that it's pretty darn rare. I'm not sure that I've actually seen it, but I do think that it's tested for by both in vitro and MNG. It's interesting. Mitochondria actually have their own small amounts of DNA within them. That's kind of weird, but it's kind of cool, right? So mitochondria, you might know, are the powerhouse of the cell. It's where all of our energy that are used by our body, are made in each one of our cells. A lot of our cells, like muscle cells, have lots of mitochondria, right? Because you need lots of energy there. Eyeballs, lots of muscles there too, right? So if you have a mitochondrial disease, it might affect muscles, it might affect eyes, things that use a lot of energy, right? So if mitochondrial disease can have very weird phenotypes because it's like it affects lots of different systems, but it's because it's affecting the powerhouses of your cell, the batteries of your cell. And these powerhouses have their own small amounts of genetic material. And it's interesting. You get 100% of your mitochondria from your mom, because eggs are packed with mitochondria, and sperm have a lot of mitochondria in the tail, but none of those mitochondria make it into the egg. It's 100% from your mom. So if you've got a genetic disease that's linked with mitochondria, and you're a male, you have 0% chance of passing that on. That's pretty important, right? That's good to know. Okay, anyway, so I'm not going to spend a lot more time on that, but that's interesting. I don't think I have enough time to go through some of these examples of kind of draw your own. I do want to get to Q&A if we have a couple of minutes for that, but I have some examples if you are interested to kind of test your theory and know if you can kind of draw in the corner and get it. But I want to ask if there's any questions at this point to finish out time, and I don't know how much time I have, but questions, please. And I'll kind of walk toward you and I'll reiterate the question so that we can have it for the camera. Question. Penetrance. Yeah, let me go back to the penetrance slide. So the question is about penetrance. And for those genes for which there is variable penetrance, that's exactly right. You have, if the penetrance for your gene is about 70%, in some cases you won't present with disease. And in 70% of the time you will. So it depends upon the gene usually. If we know a lot about that gene, if we know a lot about that disease, we can know how penetrant that disease is. So it's a function of, usually of that gene. And it's a function of how much we know about it. For some of these rare diseases, we don't necessarily know because there's not large populations of people that are well studied. And so we might not know the penetrance of a particular gene, but it is something that exists that we've observed in nature. Other questions, please. The question is, let me see if I understand it. Can you say it again? If the father's a carrier and gives HSP to the daughter, so the father is not showing disease symptoms, and the daughter is. Yes. So that can be a recessive disease, yes. Recessive can explain that situation. That's a perfect case where I would want you to go to a genetic counselor and talk about that. Because what they would do is look at your entire family tree, that you know about it. And in some cases, you might not know a lot about your family tree. In other cases, you might. In cases of adoption, in cases of in vitro fertilization, you might not know a lot. And that's the case in my family, right? I mean, my dad was treated with Hodgkin's disease and was irradiated and was literally sterile. And I had brothers who were not genetically related because they used a sperm donor. I mean, that can complicate. That's more common in the United States. But so in this case, it's good to go through your exact family tree. It wouldn't be dominant in this case unless there's an issue with penetrance. So in this case, you're saying that a father does not have disease phenotype, but a daughter does. That's consistent with recessive, but I can't say here today that that's definitely recessive. It's consistent with recessive. It's not consistent with dominant, but that's where I would want you to go through your family tree with a genetic counselor to say, what does the overall picture look like? And maybe it's something that's more complicated that we can't consider right here today. But it is consistent with recessive, yes. I don't mean to qualify it, but I mean, it's just a fact, right, as we're thinking about the clinical situation. Please, question.
If I'm the first that I know of in my family,
Yes.
and I'm concerned about my kids, would I expect that if by chance I pass something
Very good question. The question is, should we expect phenotypes in children to follow the same kind of disease progression as parents? And I'm going to describe a pretty complicated case because with HSP, it can be complicated. Remember I told you earlier to go onto that gene reviews site that Dr. Fink wrote? One of the reasons I think that's a great resource is because it actually talks about each one of the genes in some detail. It's different with each of those genes. For some genes, you can follow that pattern very well. The progression in a child is going to be much like that of the parent. And you can see disease progression that's similar across all generations. But there's some genes that cause HSP where you see widely variable timelines, time of progression, disease severity, different types of phenotypes. So it depends upon the gene, and it depends sometimes upon the actual mutation within that gene. So there isn't one clear answer. In some cases, it'll follow really well. In others, it definitely will not. It's well documented in both cases. So I'm sorry it's not so simple, but I would look to the specifics on that particular gene to see how variable is it. In some cases it's not, in some cases it is. Does that answer your question? More questions. Mark.
For recessively inherited disease, they have to be mother and father both have to have a mutation in the same gene. Does it have to be the same mutation?
The question is, for a recessive disease, does the mutation have to be in exactly the same spot in the gene? The answer is absolutely not. There are all sorts of different mutations that can happen in each gene because genes can be very long, genes can be very short. You can actually have changes all along that gene. For some genes, there's some spots that are more commonly changed than other spots. But all that you need for recessive disease is to get one copy of a mutated gene and the other copy can be a different mutation, but if it's in that same gene, that can cause disease, certainly. It does not have to be the exact same mutation in both. It's the gene that's the unit of function there, not the actual mutation. But when you get a molecular diagnosis from MNG or from Invitae, they're going to tell you what that specific mutation is, and that might be very important later on for therapy and so forth, but not necessarily for do you have disease or not. They're just looking for, the smoking gun can be any mutation within that gene. Question? Last question. And we can have time later, even privately, but.
So I have 5 children, and we know no other family members. I'm from a big family, aunts, uncles, cousins. And Eric, she's my last, is the only one that has anything. We've not done nothing as far as genetic testing. Where did we start? And like everybody, our doctors is discouraging us because of expense.
Okay, so the question is, where do you start with the possibility of deciding about testing? And you said that your doctor is discouraging you because of cost. This is a very personal decision. There is not an answer that is correct or incorrect. And that's why I refer to genetic counseling a lot, because the power of a genetic counselor is that they equip you with information so that you can make your own decision. Now, if a doctor won't necessarily write you a prescription because they feel strongly against it, I doubt that'll happen very much. Usually they're going to advise you, but really they're going to be relying upon their genetic counselor brethren to say, we're going to empower this family to have enough information to be able to make their own decision. Looking at insurance, looking at cost, looking at the family situation, looking at what is the risk that you're going to get some information that's useful. Some people want to end their diagnostic odyssey and know what the answer is. But without a therapy, it's not always necessary to know what your molecular diagnosis is. Everyone's going to have a different opinion on that in this room, right? Me, I'd rather know than not know. But that's not a right or wrong. That becomes a personal choice. So why I'm pointing you to a genetic counselor is they're going to help give you the information to make that choice. And so there's variables as time goes on. For instance, we've got sponsors, right? We've got more choice now that's actually driven down cost that makes a huge difference in the world of molecular diagnosis. We have equipment in my lab that I use. I can get billions of base pairs when before it would take years to create billions of base pairs of results. There's just higher throughput and ability. It's almost hard to analyze that much information versus get the information. So there's a lot that's changed in the environment, and so some of this information that you would get from a genetic counselor is going to change over time because the world is changing and going so fast. So there isn't a right or wrong answer. You're asking where to start. I would say start with your physician, and I would talk to a genetic counselor about it. And I'm not a clinician, right? But that's the advice that I would give. And I'd love to hear if there's variable opinions from the clinicians in the audience. I'm seeing nodding from Sabrina, that's good. But I'd want to know if there's difference of opinion, but I think that's the right advice.